In this video we will discuss about the chemistry behind polyacrylamide hydrogel. There are different ways to make polyacrylamide based hydrogel that can be used to understand the cell mechanics at different conditions, under different conditions. So we will understand one of the polyacrylamide hydrogel chemistry, and we will also understand what are the shortcomings or problems that arises in this polyacrylamide hydrogel based method. So in this method, the objective is to create hydrogel on the cover slip. First, we need to activate this cover slip so that it can interact with this 3 APTMS. There's a typing mistake. So in order to do that, we need to wash this cover slip and autoclave it, and thereafter we can react this autoclave with the NaOH so that this surface gets activated. After that, what we can do is we can put these 3 APTMS which will interact with this silicone that are present on the cover slip, and this is what it looks like after the reactions. And these reactions are done only for like 30 minutes. And these reactions are done only for 3 minutes. This APTMS addition according to this protocol. After that, they will wash this cells to get rid of excess. They will wash this cover slip to get rid of this excess APTMS, and then they will react this APTMS with glutaraldehyde, and this glutaraldehyde reactions goes for 30 minutes at room temperature. And this is what happens in those reactions, that this glutaraldehyde, one of the aldehyde will react into this APTMS amine group, and other aldehyde will remain free so that it can interact with the polyacrylamide based hydrogel. So once this glutaraldehyde is reacted, now they wanted to react this glutaraldehyde with the polyacrylamide based hydrogel. So this polyacrylamide based hydrogel is in this protocol is a mixture of polyacrylamide, is a mixture of acrylamide, bisacrylamide, and this NHS-AA Ester. In the original protocol they have mentioned to use NHS. So if we use NHS, I will tell you why we need to use NHS-AA Ester for better results. So let us first understand what happens in this mixture of this acrylamide, bisacrylamide, and NHS. So we need to polymerize these mixtures, or we need to polymerize acrylamide. So in order to polymerize acrylamide, we need some catalyst, as acrylamide is stable in its nature, and we need to initiate the free radical polymerization reactions. In order to do that, we use this catalyst APS. So if we use APS alone, then we need to heat this APS so that it will convert it into this bisulfate radical, and then it can trigger the polymerization reactions. But sometimes this heating will also have some negative impact on other reagents. So in order to avoid those heating, what we do is we use another catalyst, that is TEMED. So when TEMED comes in contact with APS, it will result in this bisulfate radical and TEMED radical. And for our interest, these bisulfate radicals is important, as these bisulfate radicals will then interact with this acrylamide and triggers the polymerizations of these acrylamide based reactions. And simultaneously, these bisulfate radicals also helps in the cross-linking of different polymers of this acrylamide with the help of this bisacrylamide, which is also present in the mixture. So finally, this is how it will looks like. This bisacrylamide is there, and it will interact with the multiple different polymers of acrylamide based monomers. It can be seen here. And since NHS is also present there, so the NHS will also take part in the free radicals polymerizations, and it will also interact with this acrylamide to form this kind of structures which has this reactive group, that is COOH reactive group, that can interact with our ECM proteins. However, the actual paper that is presented in this reference used only NHS. However, the paper that is presented in this reference, that in the frontier reference, shows that NHS itself is difficult to take part in the free radical polymerizations or free radical reactions. So in order to make it more susceptible towards free radical uh, reactions, it needs to be modified into this NHS-AA ester, NHS-acrylamide ester, and this is what it will look like during the polymerization reactions. That is NHS ester reactive group and this acrylamide reactive group. They will form these structures using this N6 linkage, and this will then interact. This will then take part in the free radical polymerizations and then form a reactive group, that is COOH, can interact with the ECM. However, this is also not that much stable, as once with this NHS-AA ester is in the solution, it can dissociate this COOH bond within few hours. So it is essential to do our reaction with ECM as fast as early as possible. These are some of the chemistry behind polyacrylamide hydrogel and what are the problems that one can face. And one of the solution is, instead of using NHS alone, one can use NHS-AA ester. We will discuss one more method in our next video. And if you need to understand how this protocol has been carried out in lab, the original video is published along with this paper. You can refer to that paper, whose reference link is in the reference. That's all for this video. Thank you for your time.